Sir, I'll share the link for the YouTube one in the chat box now. Okay. Thank you. Good.
Okay. So, Dr. Somnath, you are there?
I cannot hear you, sir.
Oh, okay. Somnath, this is Devasis here.
Yes, sir. Actually, I am the master student of bioinformatics. I am the.
Okay, okay. Good, good. Welcome to the meeting.
Thank you.
So, Samir, you would probably talk about it up to what confidence score of AlphaFold one would be reasonable sure of a good.
I will talk about how to use structures,
Exactly.
what
Yeah.
needs to be looked at.
And there are structures. We have been using AlphaFold database pretty extensively, looking at structures of our interest. But many of them have huge gaps and other things because of it could be oligomeric or things like that.
Yeah. So I'll go through what all you need to look at
Exactly. That's
to make
nice.
sense of it.
And all these you did with some huge computing farm somewhere with GPUs, or how are you? Are
It's all done by DeepMind, so it uses Google.
DeepMind. Okay. They have access to huge number of GPUs.
They are Google company, so
Okay. Please see.
is it just the NII people or is it people from outside NII as well?
I mean, we do circle, but I had sent it to quite a few number of structural bioinformatics biology people from in and around Delhi, plus our PDB India, Gopal, and some other people in MBU. And what I could think of could be Jairam's lab in Delhi because they do structural prediction. So,
Yeah.
and usually I think seminars for institute, and because nowadays it is online, it is easier for people outside to join, provided they are interested in. So
Yeah.
Sagar has joined.
Hi, Samir.
Hello.
Samir,
How are you?
We are fine, actually, considering everything. Good to see you now, long time, yeah.
I saw you the other day. um, in the condolence meeting.
Oh, yeah, yeah, yeah.
I had to leave after 1 hour, but
Yeah, it went on for almost 2 hours actually. Yeah, but I think it was
well, it's
overall.
they,
I think it was. I think all of us were a bit affected by the whole thing.
yeah, I mean, it was shocking. Debashish told me about it. Otherwise, I didn't know. I had
Yeah,
not
I
seen.
think, I mean, only those people who are either at MBU or like those who are in close touch with somebody at MBU knew about it.
Yeah,
So I have been getting phone calls from several ex people, including like Kulaskar has been, I mean, in fact, first two calls, because I keep my WhatsApp calls mute. So I
yeah.
missed, but then yesterday he again called and said that, he didn't know and all. I said, yeah, naturally, how will you know? It was only, I mean, the last year when he had his heart angioplasty, we all thought that is a very common, now, I
So
mean, in
that I
yeah,
know.
it's almost like an OPD thing they do. After that, when he was still not coming to the department, and I heard, I mean, we came to know that there are other complications have developed. So,
Quite shocking. That
yeah.
59.
But what was most shocking was, as I mean, nobody really knew what happened, but I think that's why Raghavan mentioned, if you heard Raghavan,
Yeah, I heard him about
that
diabetes.
undiagnosed diabetes can cause so much
Diabetes is
problems. I
quite
think was very
serious.
shocking, I think, for most people. So these structures are all now available, is it? These model structures on the thing?
So there are about 365,000
Yeah,
structures.
exactly.
I'll mention plans as well. So by
Yeah,
end
yeah.
of the year, there will be about 130 million structures.
Oh God. Okay.
Yeah,
So
it's going to change.
it's double the job now for you people.
Oh yeah, but it's going to have such a big impact.
Mm,
There are already so many other things people are doing with it, um, and successfully in many cases.
mm.
A lot of analysis needs to happen.
Yeah, yeah. So are they going to put out the actual algorithm or whatever AI tool or whatever they've developed? Or
Have the source code is available. There are Google collabs that you can use as well. People have already taken their code and changed and added things, and
okay.
are other Google collabs as well. Yeah, so I mean, that was the question right after CASP, what they are going to do.
Mhm.
And so we started working on it in a April, around
Mhm.
middle of April, we
Mhm.
started working on the database part,
Mhm.
and July launched. So quite tight timelines for that.
Okay. So welcome, Samir, to
Hello.
NII. Hello, I am Sagar Sangupta. So I have seen you, of course, during my time in we overlap, but somehow we have not really been in touch. So welcome to NII on NII seminar committee. Very glad to have you with us.
Well, thank you for the invitation.
So I will just give a brief outline and introduce you, and then you take over.
Yeah.
I'll take a couple of
Yeah.
So, friends, colleagues, and students, Dr. Samir Velankar did his PhD in molecular crystallography in the laboratory of Professor M.R.N. Murthy in MBU in IIT Bangalore. And from there, he moved to the Oxford University in the lab of Professor Dale Wigley for his postdoctoral work. After working on the structural elucidation of quite a few biologically interesting proteins during his PhD and postdoctoral work, he joined EBI into the work in the areas of the structural bioinformatics. Currently, Dr. Velankar leads the protein data bank in Europe in EBI, which his group has not only developed a very large number of softwares for analysis and visualization of macromolecular structures, but they also play a very important role defining the standards of validation, which is very important for the structural biology data. As many of you may know, the application of artificial intelligence by research groups has brought quite a revolution in the field of the protein structure prediction. So Dr. Samir's group in EBI has partnered with DeepMind to give a staggering number of predicted structures, 3.5 lakh predicted structures for the proteome of humans. And this is something which has their recent work, and it was published in Nature. And I leave it to Dr. Samir to talk about his recent work. His title of his talk is Structural Biology, the Impact of Structure Algorithms Using the AI Methods. Samir.
Thank you very much, Sagar. Can you see my screen?
I can
Yes.
see the screen.
Okay. So so thank you very much for the invitation. Oh, and let's just get set up. Okay. So I'll talk about the AlphaFold database and the impact of the improvements we have seen in structure prediction, and also say give a little bit of background, and then talk about the AlphaFold method at a high level, then the database, and after that talk about to use these structures, and then what impact this will have on life science research in general. So just as a background, my team at EBI, we manage multiple resources. We are partners in the Worldwide Protein Data Bank. So we manage all the depositions to PDB from Europe and Africa. We also have a data resource called PDBE Knowledge Base, where we collate annotations based on these structures and display them. And recently we worked on the AlphaFold database, which I'm going to describe. And at the end, I will say a little bit about this new project which we launched this month called PD Beacons, because it's relevant to the AlphaFold story. So in terms of understanding biology, macromolecular structures have played a vital role, and that is evident from the number of Nobel Prizes, the amount of annotations you can derive based on just a limited number of structures we have in the PDB. So my team is mainly focused on collecting the macromolecular structure data, so as part of PDB and now AlphaFold as well, and adding biological information to it to help our users derive knowledge from both the structures and the associated structural and functional annotation. So this is our mission as a team here at EVI. Sorry, I'm hearing some. Is that a question or? Okay, so this whole thing started with quite long ago in 1971, 50 years ago, when structural biologists came together and established the protein data bank. And this was the first open access digital data resource in biology, quite visionary, I think, in those times. And it's a single global archive for experimentally determined 3D macromolecular structures and associated experimental data. And why this is relevant is because immediately after that, in the 80s and 90s, there were quite a few things that research papers that came out that form the basis for structure prediction. So one of them, very early on in 86, was the relationship between the sequence conservation and the structure. There were also then prediction by homology modeling, and there were quite a few programs that were written. But even before PDB was established, it was quite clear that availability of these structures is going to help in homology modeling. So the first ever homology model was built by hand in 1969, and this is the paper. You can have a look at it. And then in the 90s and after that, there were lots of software as well as data resources that were created to provide access to predicted structures. Around same time in 1990s, it was also quite clear that coevolution of residues can be used in structure prediction. Or what was actually understood was residues that are in contact evolve. And so you can infer contacts based on this coevolution information. And these are a couple of papers from 1990s where this all started. And what also helped was this established in CAS by John Mould. So there are two things that are absolutely necessary for where we are at the moment in terms of the structure prediction. One, having data, and that was available in the PDB. And second, having a way to measure progress, and CASP provided that second assessment of progress. And it was systematically run every 2 years to develop assess the development in the structure prediction methods. And at the same time, over the last, in the 90s and in early 2000, there was explosion in public data. So both from genome sequencing, you had then lots of sequences available. They were then put into UniProt with both the sequence as well as annotations available for those sequences. Today, we have about 220 million sequences in UniProt with additional annotations. The PDB's growth was slightly slower. We have about only 181,000 structures in the PDB, and which covers less than 1% of the UniProt. So we have only about 55,000 unique UniProt accessions in the PDB, although recently with improvements in EM, we are getting many more structures as well. So in terms of availability of structures, we were quite limited. And that meant that structure prediction was still relevant. And in November 2020, we got to a stage where the predictions from AlphaFold were comparable to the experimental structures. So these are based on these GDT scores, and anything above 90 is comparable to what you might find in the PDB. And then there was a big news about how this is going to change everything. And one thing is for sure, that this established that AI can make a big difference in terms of life science research. So this is just showing the method, the overview of it, and I'll just give an overview of these three parts at very high level. But this was the most complex piece of AI that DeepMind developed, and kudos to them that they have given every detail of how this method works in their Nature publication, which has 62 pages of supplementary information and describes every bit of this network in detail. So what are the inputs? The input to the method is the multiple sequence alignment, and this is created using standard tools that we have available and the public databases. So UniRef90 is basically a database of or clusters of UniProt sequences, where the clusters have sequences where each sequence is 90% or more sequence identity to the reference sequence. BFD and Magnify are metagenome sequences, and that's another, I think, big step forward in terms of availability of sequences. So as a rule of thumb, want to have multiple sequence alignment where there are at least 30 sequences and ideally more than 100 sequences for AlphaFold to derive a good quality structure. So the input sequence which you put in is used to create an array of numbers. So these are basically residue pairs. And then you can also provide templates to the pipeline. However, often this produces accurate predictions without template. So templates are not absolutely necessary. Template information is quite useful when your multiple sequence alignment doesn't have enough information in it. So the number of sequences might be very small, or they may not be diverse enough to provide the information on covariation. The second part of this method is the brain behind this. So this is called evoformer block. There are about 48, basically. You can think of it as a loop. And what it does is it takes the MSA representation and the residue pairs, and it updates it to derive the information about the relationship between the residues in the query sequence. So at the end of this process, the pair representation, I did this first row up here of the MSA is repeatedly updated and contains then a lot more relevant information about structure and the contacts between various residues. The final block is creating the actual 3D structure. So the structure module basically predicts rotation and translation for every residue. So if you read the paper, it's a gas of residues. So every residue is kind of independent, and then this is put in place by rotating and translating. And a small network also predicts the side chain angles. And the final structure then is run through a relaxation process, although it's highly constrained, and it's not a relaxation in the sense of refinement as we know. There are two additional output apart from the structure, and we will come back to these because these are really important, or as important as the structure. One of them is the individual residue scores. This is the predicted quality score for individual residues, and this other predicted aligned error, and I will explain this in later slides. But these are basically the outputs, the structure, the predicted quality score per residue, and the predicted aligned error. And all three are equally important when you want to use these structures. So what are the limitations on this process at the moment? So one limitation is that this information is available for individual protein protomers. So it's not complexes, although there are already lots of software available, Google Colab, where you can make home complexes. And in some cases, the single chain prediction, because it's not complex, may correspond to the structure adopted in a complex, but it's not a given. And the missing context from surrounding molecules may lead to uninformative predictions. And we can see that where it produces these ribbon-like structures. And then if you go to PDB, for example, you might see a structure there in complex with another macromolecule. The other limitation is it doesn't have any information on protein dynamics. And as we know, proteins are quite dynamic. And the 5 predictions it does are usually very close to each other. In some cases, they may produce multiple confirmations, but we have to do a systematic analysis to understand whether there is any protein dynamics information if we get these 5 independent predictions. So AlphaFold is expected to produce a low confidence prediction for regions that are intrinsically disordered or unstructured. And there are already a couple of papers on bioRxiv where they have done this analysis and shown that the AlphaFold predictions for unstructured regions are really good and better than any current method we have for predicting disordered regions. The other question we get is, can we use AlphaFold to predict the effect of mutations? And AlphaFold is not trained or validated to do that. And what that means is you cannot use just change one amino acid and create a new structure and look at energetics to see if it is destabilizing. So that's you cannot do. At present, there are no ligands in any of these structures. So it does not make any predictions about any non-protein components. So no cofactors, metals, ligands, etc. And so that you have to keep in mind when you want to use these structures. And it also doesn't have any information on post-translational modifications. So which is another important part of any protein function. So overall, it may or may not lead to a hypothesis based on structure, and any hypothesis you get, you need to test it further by experimentation. Okay, so that is the kind of overall information about the AlphaFold method. Now I'll go over and describe the AlphaFold protein structure database and how we are providing access to all the AlphaFold predictions. So the decision to establish this was made on the 30th of March this year, and we defined certain principles based on established FAIR principles that we wanted to follow. So clearly define license, assign unique and persistent accessions for each of the models, add as much data provenance and associated metadata as possible. And I'll come to this. Use a formal, accessible, shared, and broadly applicable language. I have a really well-defined format for describing all the data. And where possible, use controlled vocabulary so you can then use these structures computationally. Include a qualified reference to other databases like for example, UniProt, and have an access protocol that is open, free, and universally implementable. So we provide access to this data both using HTTPS and FTP protocols. And we also decided that the launch should coincide with the Nature publication on the human proteome. Okay, so after that, we started planning, and this was the first kind of plan we made for launch. So provide data in PDB and MMSIF format. And I just want to highlight that PDB doesn't comply with FAIR principles, but it's widely used. And so we make that available, but it's very minimal. And we will not add any information to this. All the additional information will go into this MMSIF format. And so I will encourage you to use the MMSIF format from the beginning if you are going to start using these structures. We decided that we will provide bulk download options. There are some limitations on it, and I'll come back to that. The whole database is set up on Google Cloud. We plan to have web pages for the database, individual entries keyed on UniProt accession. There is an API behind all this, so we can expose that, and it can be integrated into analysis pipelines, and a basic search system, given that we had only a couple of months to do this whole thing. And we were also working with obviously my team, as well as UniProt, PFAM, and Interpro teams to integrate the AlphaFold structures into those resources. So on the 22nd July, the human proteome paper was published. And with that, we launched the database. So what are the kind of big achievement here? So one of the most important things is we now have predicted models for almost all proteins in human proteome. It covers more than 90% residues. And about 58% of these residues have this PLDDT. So this is the predicted local distance difference test score, more than 70. And we will come back to this, why this is important. Now compare that to what is available in the PDB. PDB has about 40% proteins. About 16% of these are full structure of the whole sequence. And so only about 17% of the residues are covered in the PDB. So we got a big jump in terms of from 17 to this 58%, where we have really good predictions. And in addition to that, given that about 1/3 of the human proteome is either disordered regions or intrinsically disordered proteins, this is a really good number of residues that we have predictions for. So the AlphaFold database itself is a collaboration between EMBL and DeepMind. All the structures that are available now and will become available in the future are open access, and the license is CC BY, which means you can take all the structures, use it as you like. You just have to cite the paper. The data is available for bulk download via FTP. And now we have more than 180,000 unique users since launch in the last 1.5 months. About 4,000 or 5,000 unique users every day. So for the 21 model organism that we covered, about 365,000 structures, what structures are predicted? So all sequences which are between 16 and 2,700 amino acids have a single structure in this database. At the moment, we have only made one prediction out of the 5 independent predictions available. but we are in discussion to see how we can make all 5 available. And I want to explain this accession a little bit because we get a lot of questions about this. So similar to a PDB ID code, this is the AlphaFold DB ID code. And it has the pattern AF hyphen P12345, which is the uniprot accession. This much is clear, and we get questions about what is this F1 to N. So let's go back to this 16 to 2700. So what happens to those proteins where the sequence length is more than 2700? So for those, there are multiple predictions made in a window size of 1600 residues. So you have 1 to 1600 as, let's say, AFP12345F1, then 201 to 1800 as F2, etc. And so, for example, for Titan, you can imagine there are many, many, many models. And each of them, they have a specific accession. We also made sure that we version this. So the model file will have a version. At the moment, obviously, everything is V1. Both the PDB and MMSIF are versioned, and this predicted aligned error is versioned as well. The web pages themselves have basic search systems, so it allows you to search using uniprot accession ID, protein name, gene name, and organism. And this is limited because we had only 2.5 months to basically get all this work done. We will implement more functionality in the future. On the pages, we wanted to make sure that there is a clear indication that the structure is shown is a prediction and not experimental structure. So you see that at the top. Then it allows you easy download access. It also provides some basic information about the protein, and it highlights if you have any information in the PDB for this particular protein. And it links back to PDB knowledge base where you have all the information about all the structures from PDB for a given protein. And then the structure itself is shown in 3D viewer, and it's colored by this PLDDT score. And let's go into that into more detail in the next couple of slides. And then just below this, there is this predicted aligned error. As I said, it is also accessed through multiple data resources. So you can go to any of these data resources and access structures from AlphaFold-DB. Okay, so what are we going to do next? Something under consideration in terms of data is removing the signal peptides from the predictions, making all 5 independent predictions available for every protein, and additional metadata available for each of them as well. So including possibly multiple sequence alignment, although this is very tricky given the size of it, but we will look into that. Information on the templates that may have been input and may have been used by the network, and the quality criteria. We may add additional quality criteria to make it easier for it to be used in different contexts. The plan is to update the data to complete UNIREF 90 dataset, so about 130 million structures by the end of this year. In terms of web pages, we will design web pages for uniprot accession with multiple fragments. So at the moment, we have pages only where there is one fragment structure. We want to extend that. Also design pages for the individual structures so that you can go into more details for each of them and continue to add more functionality. Okay, so from here, it is more, I think, about how you can use these structures. So the first and foremost thing to remember is that these are all predicted models. And as this quote says, and this is true for PDB as well, all models are wrong, but some are more useful. So you need to really understand what you can and cannot do with a given model. So two things to remember is AlphaFold was trained on PDB structure released before April 30th. So any structure you take from PDB before April 30th, 2018, and compare it to AlphaFold structure, it's going to get it right because it was in a training set. And anything we have in the database, we also provided additional templates until February 15th. So which means there is a possibility that it gets biased by those templates as well. So those are two things to remember, and I have something more how you can validate it a little bit later. So let's go through these two scores that are very, very important if you want to use these structures. So the first one is predicted local distance different test score. So this is AlphaFold predicts per residue this score. It measures the kind of percentage of correctly predicted interatomic distances within a local region. And it rewards the locally correct structures. And this is, so for a given domain, for example, this is very relevant. So if you see domain, it's very relevant that you look at the PLDDT. Right. So the PLDDT score ranges from 0 to 100, and 100 is most confident prediction. 0 is least confident, or you can't believe anything that is 0. And we use then consistent confidence bands in the color scheme that we use to display the structures. And it has a meaning that we'll come to. So here you see that the dark blue is PLDDT above 90, then slightly less dark is this blue 70 to 90, etc. And both these are available in the B factors as well as in MMSAFE. There is a category you have this information in both the global value as well as individual residue local value. So the global value is missing in the PDB. So how do you use it? You can look at the structure and identify domains using this color scheme, which is based on PLDDT. So anything that is less than 50 is not a good prediction, or the AlphaFold is not confident about that prediction. But it has been shown now, and there is more work being done on this, that this is a really good predictor of disorder. So this part is all disordered. This is a good domain. And then within a domain, you can look at which residues have really good confidence score. So what is observed is if it is above 90, then even side chains are really properly placed. So you can use the PLDDT to both identify domains as well as within domain identify regions with high confidence. So what are the pitfalls of just using PLDDT? So here I have a couple of structures, and as you can see, there are multiple domains here. And PLDDT is not about how good the placement of different domains is with respect to each other. So it doesn't give you information about the relative positions. So that is something that you still have to worry about. So for example, here, the domains themselves are really good. And because they are not next to each other, fine, you can conclude that these are two independent things. So the beads on a string. Whereas here you have multiple domains, but they are all close to each other. But their PLDDT is not going to tell you whether the relative position is predicted with high confidence or not. And for that, then we need another metric, and that's the predicted aligned error. So AlphaFold's predictions of its position error at residue X. So this is what we are trying to provide in this PAE plot. So if you kind of superpose the true structure with AlphaFold prediction on residue Y, what is the error you observe at residue X? So for each residue Y, you are going to get error for every other residue in the structure. So let's go through some examples to make it clear. So this is a PAE plot. And here it's quite clear that there are two domains. And as you can see that this is really dark green, which means the predicted error in the positions is very low. And so you can identify this as one domain and this as the other domain. Right. So what about two residues in the given domain? So let's look at these two residues. So you go to PAE plot and you see that for these two residues, it's dark green, which means the relative position of these two residues is predicted with high confidence. And so you can be confident that this is good. What about the relative position of residues between these different domains? So again, you look at the PAE plot. And here you can see that it's white almost, which means the error is quite high. And so the relative positions are not confidently predicted here. So these domains might be doing all this dynamics. And what you have in the coordinates is just one of those different conformers. So just to summarize that, here you have multiple domains. And as you can see, really, these are all independent domains predicted with high confidence, but there isn't enough confidence on the relative position of those domains. Whereas here, you can see that there is some confident prediction about relative position of some of these domains as well. So use the structure, the PLDDT, and the PAE together to really make sense of it. And just to emphasize that point again, here is a biological example. So this is a Arabidopsis protein. It's involved in protein export, and you can see the structure here. And you can see that there is a pore here. And the N-terminal helix is in that pore. So you might think of that it may have some functional role. But if you look at that helix itself, the PLDDT is quite low. So it's not really a good prediction for this region. And if you look at the PAE plot, there isn't good interaction between, or the relative position of this helix with respect to the rest of the protein is not confident either. And so don't conclude, for example, that this has some functional role. So this is quite important, and make sure that you make use of it. So how do we validate this? One way to validate it is to look at new structures that we get in the PDB, compare them to predictions and see how good they are. So here is one example where this is a new structure that came out. The nearest one has 27% sequence identity, but you can see the prediction. I mean, you can't tell which one is which one is experimental and which one is prediction. The other thing that is amazing is if you look at these metal binding sites or the sulfur ion cluster sites, the side chains are very well placed as well. And that I found amazing, although this is anecdotal, so I haven't seen it across the whole archive. But we can do this continuously. So every week when new PDB comes out, we can compare and and we can see if the predictions are good, and that will build the confidence in these predictions. So the Cameo team has set up such a pipeline. You can access it here, and you can every week see the new targets being compared and giving really good confidence in these predictions. Okay. So the last part, what's the impact going to be? So until now, we had a very limited number of structures available. And now we are in a situation where structure for every known protein in UniProt will be either in this AlphaFold database, or we can model it because there will be a prediction that will be 90% or around that sequence identity. So this is going to have a big impact in structural bioinformatics, obviously. We are already seeing people moving to predicting complexes of macromolecules, homo and hetero, both, and there is already a lot of Google collabs available for that. Although I haven't seen anything on the protein nucleic acid complexes yet. And now that we have really good handle on the intrinsically disordered proteins, that part of the scientific study might become really relevant now, and we need to maybe develop many more tools for this. There may be more we can do in terms of actually getting protein dynamics information out of this and understanding whether we could also predict relevant conformational states of proteins. And already there are people who are looking into this. Having all these structures available, can we then derive some information about functionally important residues? So for example, information on impact of mutations, binding sites, which residues are important for these conformationally distinct states, interfaces, and then start looking at this also from point of view of which ligands might bind into different pockets. Although you have to be really careful because these are, as I said, not complexes. So you need to understand the protein a bit more as well in this case. But this is going to accelerate structural studies a lot, so in different ways. So this is just based on all these different tweets I have seen. So improving construct design. So now that we have a handle on which parts of the protein might be disordered, you can use that information to make a construct that will crystallize or will behave well on EM grid, etc. Obviously, this is going to provide starting models for structure determination. We can take low resolution maps and start looking at the models and fit them into low resolution EM maps, do time resolved studies, etc. So structural biology is going to explode and have lots and lots of new complexes studied. But it's also going to help with integrative hybrid methods. Here we are looking at huge complexes like nuclear pore complex, and having models for each individual component is going to make a huge difference to these methods. But if we think about it, maybe the experimental methods which provide sparse data, so not necessarily high resolution EM or these integrative methods, etc., but just provide enough information to give some actionable data to test hypotheses. So chemical footprinting, hydrogen deuterium exchange, or FRET, these methods alongside these predicted models can start providing us with real biological insights into the function of proteins that we are looking at. So how can we facilitate access to all these structure models? So predicted models, experimental models, etc. And so this is where we started a project 2 years ago called 3D Beacons in collaboration with all the different data resources and now including AlphaFold DB. One of the questions that is being addressed in this is how do we assess quality of all these different models that are coming from different methods, and how can we rank them? So the idea behind 3D Beacons is to improve coverage of sequence space by giving access to all these different data models from different data resources. So create a common API specification that gives access to both experimental data as well as predicted structures and the associated annotations in PDB knowledge base, and provide the quality assessment. So we already have some prototype, and as I said, we will release this month. There is a common API that you can look at on GitHub, and the web application will be available as well. So overall, as I said, in my team, we want to go from molecular data to knowledge, and we are creating the necessary infrastructure that provides access to structures, integrate it with annotations, and then create APIs and web pages and web resources, analysis tools that provide access to user community. So these are the 4 resources that we work with. And with that, I'd like to acknowledge the people who were involved in the AlphaFold project from EBI and from DeepMind. And I'll stop there and take any questions. Thank you.
Sagar, mute probably.
Sorry,
Sagar, unmute your.
Over it on this screen. On his. You can hear it. No? What is happening?
Sagar, can others hear? We can't hear you here.
Is Sagar audible to everybody?
Yeah, I can hear you, Sagar. Although it shows you on mute, but I can hear you.
Sorry. Why we cannot hear Sagar? Okay,
So maybe people can write questions in the chat, or somebody else can triage them. I don't know.
Yeah, there was a. Yeah,
okay,
anyway.
Okay. Yeah, we are solving the problem. Yeah. Okay. Sagar, can you say?
Yeah, now it's fine.
Okay.
Now we can unmute.
Yeah, sorry about that.
Hello, Samir.
Oh,
Samir, this is Vichitra here.
oh, how are you?
Good, good. How are you?
Vichitra, go ahead with your question if you have.
Yeah. Samir, can you hear me?
Yes, I can.
Yeah, it was a wonderful talk, and thank you for doing a great job.
Thank you.
I have many more questions, but I will limit myself with 2 questions if Sagar allows me.
Go ahead with the question, Chitra.
Yeah, so my first question is, in your view, do you think the protein fold space is largely over or covered? Because nowadays we do not come across new folds.
I think that is a really good question, and that is being analyzed as we speak. I'm working with Christine Orengo's team to see if any of these predicted structures are completely new folds. But that analysis is not yet complete.
And sorry, please go ahead.
Yeah, so we have to wait for that analysis to be completed so we can see if we find completely new folds.
Okay, with Sagar's permission, I have another question. That the structure that you have predicted using your model system, and how many of those predicted structure eventually been elucidated experimentally, and how much correlation you find between the experimentally determined structure and predicted structure, if you have any examples?
So I gave a couple of, so I gave one example, but I have many more, that it is absolutely amazing how this works. Let me just share my screen again. And
Yeah, I'm sorry, I may have missed. Yeah.
So this was a completely new structure that came out after we released the database, and it was deposited before AlphaFold thing, which did have really good model in the PDB, but you can see that the overall structure is fine. There isn't much difference except in the loop regions, but even the side chains for structural metals and things like that are really good. And I can give you a couple of more examples like that. So here you can see, for example, this is a complicated zinc finger with 3 zincs next to each other and lots of different cysteines, but they, again, all the side chains are in exactly the position they should be. For zinc, here is another example where it's a homodimer, and red is the prediction, and again you can see that it's perfect. You can just put the two together and make the homodimer. But what I was really impressed by was this structure. So here we have a DNA bound structure in the PDB, and you can see that this brown is the predicted structure, and it has all the loops and everything else in exactly same place around this DNA. So what we are finding, and again, this has to be done in a systematic way across the database, but what we are finding is for new structures, the predictions are really, they match the experimental structure. You really can't tell the difference except in the loop regions and in the disordered regions.
So can I go ahead with the question?
Thank you. I am done, Sagar.
Sagar, you're not audible, so I presume that I can go ahead. This is Pushkar.
Yes, you go ahead.
Yeah, a quick question. You talked about PLDT scores. While it is understandable for disordered region, they're most likely less than 50, but there are instances where very well-defined alpha helices are also shown to be less than 50. And how does one interpret? And then of course, but surprisingly, in structures of homologous proteins, they actually are alpha helices in some cases.
Yeah, so that's a really good question. And just if you attended the webinar we had, Balint from Heidelberg is doing analysis on.
So this is the structure, like for instance, if you can see.
Yeah, intrinsically disordered regions. And what he found was even in intrinsically disordered regions, there are short stretches of helices and things like that predicted. And if you then look at the complexes of that particular protein with another protein, these parts are actually helices, and they interact with the other protein. So where you have intrinsic disordered, in some cases, and again, this has to be done again systematically to understand under what conditions AlphaFold actually is predicting the secondary structure in for the regions that bind in complexes. But that's one of the things he has found, and he's analyzing that further.
So Dr. Samir, then at least for speculation or hypothesis, would it be a good hypothesis then? It's quite possible if the situation is what I just mentioned, it's quite possible that it may be part of a homodimer or interaction with another protein, which basically may facilitate that secondary structure.
That's the hypothesis that Malint and Norman Davey are actually testing at the moment.
Thank you very much for your response. Thank you.
I don't know whether I am audible or not.
Sagar, you are now. Now you are.
Okay, that's great. Rajesh.
Yeah, I think if you can. Hi, Samir, it's good to see you actually after a very long time. And
good to see you.
Yeah, good to see you doing well. I think it was fantastic. So I think great paper. I mean, I haven't really for a long time come across something, that is as prophetic as it can be now. So now that has all the structures intact, can you start predicting functions for many of those on which we are unaware of what they actually do? Is that feasible in a timelines that you allow biologists to almost assign functions for proteins that are very, very difficult to figure out?
Yes and no. So, for example, I gave an example of the binding sites. Now, because these are individual chains, one thing that happens sometimes is the pocket. If you start looking at where the pockets are, you may not necessarily get the right pockets, and sometimes you get pockets where if you then have this protein in complex, you won't have that. So having said that, because as I said, some of these side chains and things like that are really in good place for cofactors and metals and things like that, at least from what I have seen, we can start looking at predicting which ligands bind, structurally important ligands. That's first thing we can do. In regions where the PLDDT is very high, we can start looking at mutations as well, and whether those mutations then are close to these kind of predicted functional sites, etc. So you could definitely get some hypothesis given all the other kind of quality metric support that hypothesis, and then do experiments based on that. And that's what is exciting, that every protein will now have a structure to go with it, more or less. So you can really get a lot more out of it than just having a sequence.
Yeah, thanks.
Dave?
Yeah. Samir, nice talk. The question I had was either your group or anybody in DeepMind has done this exercise instead of using the structures up to 2018 for training. If you used only up to 2010 or 2008, what would be the outcome on the prediction accuracy or things like that?
I haven't asked them if they have done this, but it's a good question. So in a way, what this is doing is it's a good mimic of all the different packing you find in the PDB. So what you are asking is, have we seen new types of packing after 2010? And one way we could maybe look at it is how many new domains we have, just as a indicator, after 2010. But I haven't asked that question to them.
Yeah. Another, if I have just a quick question, I think one of the success for them was to use this their BFD or whatever big fantastic huge database of sequences. So are
Yeah.
they using it for contact prediction, which you explained, or is there any? I mean, including meta, I believe they have used there.
Yeah, so it's basically to get better multiple sequence alignments. So to get more depth in multiple sequence alignments, so that they can predict with higher confidence this covariation.
Okay. Yeah. Other scanners.
So Samir, there is a question in the chat box. Does the AlphaFold prediction close to X-ray or NMR structure?
It's a good question, and you would expect me to say, yeah, it's close to X-ray, because obviously, you know, the training set has a bias to all the X-ray structures. But having said that, the CASP competition last time also had NMR structures, and AlphaFold did really badly on those. In terms of comparison of AlphaFold prediction and the structure in the PDB, there is a new paper from Guy Monteleone, either on bioRxiv or may have got published now, I don't know, where he has compared the raw data from NMR, so chemical shifts, NOEs, etc., with the prediction and with the PDB structure. And actually prediction did better in terms of correlation with the experimental data than the structure in the PDB. So you can make out what you will of that, but it's a really good model, even for NMR data.
So that was from Dr. Gopalan in NII, and Professor Manju Bansal has put up her hand.
Hi, Shamir.
Professor.
Yeah, I had a question, because you did say that some of the modeling, they come out with multiple models, like 5 models or something, but what they put out is only one.
Yes, at the moment we're only putting out one, because we had a really tight timeline to start thinking about all multiple models and things like that.
So is it likely that in near future the others would be available?
It's likely. This is what we are discussing at the moment. What do we do next?
Yeah, because the reason I ask is that because, I mean, my immediate interest is on protein nucleic acid complexes. And there, from what we've looked at some of the complexes, and one of my postdocs, in fact, in this PDB project that we have, she's been examining, is that, you know, when the protein, same transcription factor binds to different DNA sequences, especially if flanking sequence is different, then very often looks like the protein undergoes a small, especially the side chains, you know, they adjust themselves to interact with the slightly different sequences. So I was just wondering whether intrinsically, if people do that kind of modeling from AI also, they're able to see those minor differences.
So I did ask this question, and this goes to what I was saying about dynamics. It may be local or global, and they have done quite a few experiments, but the network was finally trained to produce kind of one structure. And so it's not trained to do the dynamics part as such. And this is why I said you need to really do a global analysis to see if we take 5, does it really provide enough kind of variation in those structures or not?
No, but when you say dynamics, do you mean like a molecular dynamics type of, you know, like in the thing, NMR thing, or just from the AI inputs can we get, when you get multiple structures, how reliable would they be? And how do they choose the one structure that is closest to the known structure, is it?
Yeah, so that's with the highest global PLDDT.
Okay, so these are these.
So but all the others are not that far, but they may be slightly different conformations. So what you are saying, the AI network is not trained to do what you are saying, that you create slightly different structures. It is trained to really get one good prediction for the final. But they have done quite a lot of experimentation on, can you actually get multiple structures? And that's a research topic that they still might work on in the future.
Okay, thanks. It was great talk. Great hearing you. Yeah.
So there is a question from one of the students, Ahmed. Some of the structures have tendency to domain swapping. Can we see this in AlphaFold?
Well, you won't, because it will only have one of those chains. And you won't see the domain swapping. What you would see is the domains being in slightly different place. But if we get to prediction of complexes, so homodimers, for example, where we have some domain swapping examples, then we might see some of those if we are successful in that. But at the moment, you won't be able to say anything about domain swapping.
There was an earlier question from another of the student that can AlphaFold predict oligomeric state of the protein? Right in the
well, there is a Google collab from Sergey, I forget his name. It's called Old Collab or something like that, where he is looking at how we could get multimers, so prediction of oligomers using AlphaFold, but it's not validated at the moment. So that's still a research project.
So Dr. Devendra Sehgal has asked, how far is AlphaFold away from predicting protein interacting partners?
It's not at all trained to do any of that. It's really just trained to get the structure of the protein. Predicting protein interacting partners is a very complex thing, especially for eukaryotes. So it's not working in that space at all.
I think most of the questions in chat box we have.
Sagar, Devastuti has some question. He has raised the hand, I think. Devastuti.
Please go ahead.
Can you hear me, Professor? Okay. Thank you so much, Professor. It was a very nice talk. I work for PTB India. So as part of our collaborations, we do collaborate with a lot of experimental labs. And the one question that many of them have asked me is, especially when you talked about PAE, and we are not sure of the domain interactions. And the one thing that they want to do is, from the structure, they want to go ahead and mutate some residues. So is there a quick way that I can do something with a poorly described domain interaction so that I can give them an indication about whether you go ahead with this prediction or don't go ahead with, mutation, or don't go ahead with it, because it takes a lot of time and effort to do mutations, right? So do you think a quick simulation of advanced molecular dynamics, or what is it that I can do to help them better with this?
I don't know, because if you do simulation and the domains come together, that won't mean anything, because it's not necessarily any meaningful state than if the prediction says that the domain interactions are not predicted with high confidence, I would think. Maybe somebody else has a view on that, but so it may be for various reasons, right? It may be because this protein interacts with another protein, and those two domains then interact with that protein in a different way. So there can be many reasons why those two domains are not next to each other. And so based on predictions, and if the PAE says that it's not next to each other, that I don't know how you will have more confidence if you just do molecular dynamics and if you see a state where those two domains are together. I would rather look at databases like Intact and other databases where they may have mutation experiments or biochemical experiments that give you more information on interfaces.
Thank you so much.
I think there is Priyank also who has raised a hand. Priyank.
Thanks for the talk, and it was wonderful talk. I just wanted to ask two questions. First of all, you said about the predicting the. Am I audible?
Yeah.
Yes.
Yeah.
Yes. Go ahead.
Yeah. So first of all, you talked about the protein-protein structure, protein-protein interacting partner and all, but uh, 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 a month back only a paper in Science came about Rosetta 4.
Yes. Yes. I can guess what you are asking because you are breaking up. So both Rosetta Fold and
that it can
AlphaFold
predict the
are very similar.
protein interaction and protein. So
Yes. So let me answer that. So both Rosetta Fold and AlphaFold are similar method. Actually, Rosetta Fold is based on what AlphaFold or DeepMind presented at CASP. So obviously, you can always find examples where AlphaFold will also give you right complexes. Already there is a bioRxiv paper on protein peptide complexes using AlphaFold, and that was benchmarked. There is, as I said, a Google Colab which uses AlphaFold to predict complexes. The point I was making was that it's not benchmarked and validated that the complexes it produces, what confidence we have in those predictions. You can always find examples where it works and you can display and show a couple of examples. That's not a problem. Both Rosetta Fold and AlphaFold need further validation in terms of their predictive power for complexes. And I think the science paper says that as well, that these are some examples and it then needs a lot more further work to make it into kind of system that you can rely on for predictions.
and another thing I wanted to ask is because there's a lot of roles of intrinsically disordered proteins or IDPs right now in biology, and so what about those structure, whether it can predict such intrinsically disordered proteins also or not?
Yes, so anything below 50 PLDDT is shown to be a good predictor of disorder. And the previous question about having small stretches of helices and things like that in those regions, as I said, they sometimes are the regions which are interacting. So, but a lot more work needs to be done again to kind of validate that and benchmark that.
Okay, thank you.
Thanks, Priyank. So there are a couple of more chat questions which are there. So one of them is from Dr. Madhulika. Is there a differential confidence in predicting different kinds of domain? For example, she's asking about DNA binding domain.
I mean, I don't know because it's not yet. So nobody has looked at this across the whole database, but I don't think why that should be the case. that there is a different confidence. Obviously, membrane proteins is a different ball game altogether. Although I have read a paper in bioarchives that even those many cases have really good predictions. But I don't see why different domains should have different predicting power. Unless I misunderstood.
Okay, so from there is another clarification. She is asking whether AlphaFold used any energetics or thermodynamic parameters.
So as I showed, the last step after structure is constructed is a relaxation, but it's heavily constrained. So it's not really a proper energetic or thermodynamic parameters used in prediction at all. It's really just to relax the structure locally in places, so it fits the criteria for good Ramachandran angles, etc., good torsion, etc.
So I think there is no other hands up or any questions which are coming up. So let me just ask one clarification, maybe trying to understand how much AlphaFold coming into the in vivo situations for us cell biologists. We are obsessed with post-translational modifications, which probably is not at this moment taken into account. So is there a thought process of something like integration with a system biology type of situation where the predictions of the cues come in and then it becomes a much more probably almost predicting the functions of almost every protein within the cells by AI?
Yes, so this is the in situ structural biology efforts with tomograms and volume EM, etc. And these predictions are going to be really amazing for them as well in terms of fitting these models. And as we get better at predicting complexes, those models into the in situ EM and correlative EM, etc., light and EM microscopy. So yeah, you will start that kind of thing, seeing that kind of thing in the next couple of years, probably.
But fantastic for giving us a preview of what is exciting in front of us, Amit. It was really a privilege to hear you, and very glad that you came over here virtually and gave us a talk. And I hope next time you come to India, you will be visiting us in person, and it will be a pleasure to have you here in NII.
Well, thank you very much for the invitation. It was really a pleasure to talk to you all.
Yeah.
And see some of the old friends. I haven't met Rajesh for now many years. Okay, thank you.
Thank you.
See you.
See you.
Bye.